today. Uh, okay, so let me introduce myself. Uh, I am uh, first welcome to this webinar, and it is on on clinical trials. And myself, Rahul Bhattacharya from Calcutta University, Department of Statistics, and I am working on this clinical trials field for the last 10 to 15 years. And actually, in these hard days, we all go through the words like clinical trials, vaccination, efficacy, randomized clinical trials, and so on. And so in this webinar, I will try to address some issues related to these terms and some development regarding this vaccination trial and give some insight. Although the topics of clinical trials involve rigorous mathematics, but keeping at the view of just the participants from diverse fields, I will keep my lecture at a very popular level without involving any type of mathematics. So I will start now, and so can you see the presentation now? Okay, so we are just about to start this. So I'm just for the timing, I'm just keeping my camera off and then proceed further. So first thing is the topic is clinical trials, as we have said that what is a clinical trial. So the organization of this talk is like that. What is a clinical trial? What are the different terms involved in it? How it goes? And then finally, I will discuss some of the issues, some of the upcoming issues for a clinical trials, so that you can understand the basics of clinical trials and just identify different issues and different deficiencies for the going on for the clinical trials just now going. So the first thing is what is a clinical trial? Actually, if you go through that word that is clinical trial, you will find that this clinical trial, the word, it consists of two words, clinic and trial. It is this word trial is originated from the Anglo-French word trier, meaning to try, and the French word is clinic, refers to the practice of caring for the sick at the bedside. So taking this together, the word clinical trial literally means the activity at the bedside of the sick. However, this definition is obsolete, and nowadays clinical trial has some new dimension and new definition. So nowadays, a clinical trial is a research study with human volunteers to answer specific health questions. So nowadays, clinical trial is a research study. Lots of money is involved in it, and human volunteers are also involved in it. And we have some specific health question, just like the current pandemic. So we have the question whether this treatment is doing better in this pandemic situation or not. So to get the answer, we have to conduct some clinical trial. So definitely it is a research study and it will involve human volunteers. Otherwise, it will not be possible to get correct answers. And and most importantly, carefully conducted clinical trials are the safest and fastest way to find treatments that work in people and new ways to improve health. So these clinical trials are aiming at the mankind to develop the health system and give better treatments or better drugs for some disease, and this is to just a way gives a new ways to improve health. Now let us start from very origin of clinical trials. So you know, experiments in medicine are as old as medicine itself. In fact, no medicines can be developed without any clinical trials. So whenever you find some medicine, uh, in behind there must be some clinical trial. Now, the form of the clinical trial may differ from the nowadays trial, but the idea is like that. You have some research study involving human volunteers, and there is some medicine or uh, some promising treatment of a promising substance, and then you get the medicine. So the first example goes to nutritional experiments in the Old Testament. So it was about 800 before Christ time. So the trial was very simple. Two groups of patients were there. One group was given the conventional vegetables and drinks, and the other group was given the food prescribed by the king. So here, king is the experimenter, and the experimenter wants to know whether his prescription or his treatment, that is the his prescription or his prescribed foods are better to are better to keep people healthy or not. So this was a basic assumption for the trial, and the final observation was that at the end of the 10th day, the subjects following king's prescription looked healthier than the subjects of the other group. So this means it was a clinical trial, not conventional clinical trial, but there were two treatments. So one was the conventional vegetables and drinks, and the other was some food prescribed by the king. And at the 10th day, you find that the king's prescription is promising to keep people healthy. So it was one example of clinical trial. So it was a very early trial, at least reported. Next is a little bit new. It is scurvy trial of Linz. It was conducted in 1753. The trial is like that. Scurvy is known to occur by the deficiency of vitamin C. So among a total of 12 scurvy patients, two patients allocated to each of, there were 6 treatments actually, or 6 promising substances: cider, elixir, vitriol, vinegar, nutmeg, seawater, lemon, and oranges. And since we know that this scurvy is known to occur by the deficiency of vitamin C, so we find that lemon and oranges are most promising, and the same observation was followed by the was observed by the Linz, and he find that at the end of the follow-up period, lemon and oranges came out as the most effective. So it was the most recent times clinical trial, but not in the name of clinical trials. It was for the patients, and it was to just to get some idea how to treat patients with some substance which have some effect on scurvy. Now we recently come across this type of uh, terms like treatment, intervention, placebo, control. What are these? So this is the basic question: what are these? So what is intervention? Actually, treatment, the word treatment is used in a generic sense. Okay, so it may be intervention, it may be placebo. So what is intervention? Intervention means the new drug or therapy whose effect is under study. So if your treatment is a new drug whose effect is under study, then this treatment is intervention. Uh, Next is placebo. What is placebo? Placebo is is a is an inactive pill, maybe liquid, maybe powder, having no treatment value but has some psychological effect. So if your treatment is a, it might be possible that your treatment is a placebo. Next is control. What is a control? Control, this is a standard by which experimental treatments are or some existing treatment or existing drug, and your intervention is to be compared with this one. So whenever we use some control, this means patients conditions are under control with this control treatment. That's why this name is control. So these are further terminologies. Control trial. Control trial means comparison of a new treatment with either a standard treatment or placebo. That is control. There is another term, randomized trial. So in randomized trial, allocation of patients to treatments by some objective are done by uh, using some objective and in some non-deterministic procedure. So I will come to this randomized trial later. And what is randomization and what are the utilities of randomization? I will discuss all these things later. Next is the most important randomized controlled trial or RCT. Uh, if you recall uh, the Nobel laureate Abhijit Binayak Banerjee's work, it was some economic RCT, that is RCT conducted for economical purpose. So the idea is that is an experiment performed on human subjects in which the new or experimental treatment to given one of the group of the patients while the other group receives the standard treatment or placebo. So there is some control treatment term, one active treatment term, and if this placebo is used, the trial is called placebo controlled trial. Otherwise, it is called a randomized controlled trial or RCT. So for example, we have just discussed the Old Testament trial. You can find that uh, uh, just according to this terminology, the vegetables and drinks are the conventional, that is the conventional food is the control group, and the king's prescription gives the treatment group. So it was actually a controlled trial, so comparing two treatments. Next is in clinical trials, since human volunteers are involved, so there must be some ethics. That's the most important thing in clinical trials. So whenever patients enter into some clinical trial, there are several treatments for assignment, and definitely not all are the best, or it is not possible to get every best treatment in a trial. So there must be some inferior treatment. Now, uh, so what is ethics in clinical trials? The ethics is that uh, if a patient enters into the clinical trial, you have to put the patient to the best treatment. That's how ethics can be maintained. But the problem is that before the experimentation, how do you get the best treatment? Now, there is no way to get the best treatment even just before the experimentation. So what to do? So, but assigning a patient to an inferior treatment is a serious objection from the patient perspective. So we have to be uh, choosy about two types of philosophies. So two types of philosophies are individual ethics and collective ethics. So there are two types of ethics actually. One ensuring that an individual patient receives the best treatment. So this is the individual ethics. That is, from individual patient's perspective, I will get the best treatment every time. So this is what is known as individual ethics. And the next one is evaluating therapies as efficiently as possible so that all the future patients may benefit from the superior treatment. This is called collective ethics. So the idea is that sacrifice some patients, that is, sacrifice means assign some patients to the treatment not doing better or to the other treatment arm so that we can get comparable groups. Because if you know that some treatment is doing better, there is no need to experimentation. So if in course of the trial you have some idea about which treatment is doing better, you can assign all future patients to that treatment. But there is a serious objection, because if you assign most of the patients, say 90% or 95% patients to one treatment and 5% to the other treatment, how do you compare the results? So one is getting 95% of the assignments and you get 95% responses from one treatment arm and only 5% responses from other treatment arm. So there must be some inferential issue that one is getting higher weight and one is very lower. So you have to make a balance between these two, that is individual ethics and collective ethics. So this is the main problem in clinical trials. Whenever you perform some clinical trial, conduct some clinical trial, there must be ethics, and you have to be very careful to maintain the ethics. And there is individual patient ethics and collective ethics. So being a clinician, you have to just strike a balance between these two ethics. Now, in a broad sense, a collective ethics leads to individual ethics. So if everyone is happy, then every individual is happy. So that's the idea, how collective ethics leads to individual ethics. Now, let me discuss some other things. Why ethics is, so these slides will tell you why ethics is very necessary in clinical trials. So this is, I will discuss some unethical clinical trials. So early experiments on humans have been conducted without taking care for the welfare of the subjects, who have often been prisoners or disadvantageous people, say slave, say prisoner. So they are often disadvantageous and they don't have their own consent. So they are being used for clinical trials. So my first example is that in the United States, in some times, physicians put slaves into pit ovens to study heat stroke and poured scalding water over them as an experimental cure for typhoid fever. Yes, we agree that this type of treatment is possible, but not in this form and not on the slaves. Okay, so you have to respect for the slave's position. Next one is example of control trial where one slave had two fingers amputated, uh, with one finger with anesthesia and one without. So the idea is that if you get uh, if you feel more, uh, which one feels more, with anesthesia or without? So the idea is this is a controlled trial, but obviously this is unethical clinical trial. There is no types of ethics for the involved patients or involved subjects. So unethical experiments, this type of experiments, I am just giving you two types of examples. Unethical experiments for drug development with human beings continued until the Nuremberg Code. uh, Nuremberg, you know, the concentration camp for the German people. The first international effort was to lay down ethical principles of clinical research. So these are some international guidelines. It started from Nuremberg Code of 1949, then Declaration of Helsinki regarding during 1964 to 2000, and then there was Belmont Report. In all these guidelines, uh, it was said how to conduct some experiment or clinical trial when particularly human subjects are involved. So these are some common procedures, just creating some uh, your trial is maintaining ethics or not. So this is the ethics committee. Then comes informed consent. That is whenever you take the include subjects, whether voluntary consent, whether they are giving their informed consent. That is whether the patients to be recruited are given the full information about the trial, uh, what are the pros and cons of the trial, what they get, and finally that this is research and this is not treatment. So this all types of things are uh, uh, in, are, are within the purview of informed consent. Next is vulnerable population. Vulnerable population means from clinical trial exclude vulnerable subjects unless their participation is needed for scientific reasons. For example, children, mentally impaired, individuals with dementia. Prisoners are thought not to have autonomy to give informed consent, so they are excluded from trial. Of course, students, subordinates, patients are unduly influenced to participate. So clinicians need to ensure only parent can consent for minor, and consents must be in subject's native language so that the participant can understand what is going on in this trial and what is expected from him. So this is all about human subject protection. So coming to the Indian guidelines, so Indian Council of Medical Research, that is ICMR, translated most international ethical standards to India and legislated recently. So this is now legal. They have suggested to form institutional ethics committees, that is something parallel to the concept of EC, that is ethics committee. and there is some legal regulator for United States. It is FDA, that is Food and Drug Administration, and here the legal regulator is Drugs Controller General of India, or DCGI, who provides clearance to clinical trials subject to some IEC approval. Now, so these are the basics of clinical trials. So if one is interested in conducting clinical trials, he has to follow these steps, approach to the IEC, then he will get the idea of clinical trial and consent for the clinical trial. Now, what are the different phases of a clinical trial? What happens during a clinical trial? This is the most important question. So from the development of a new pharmaceutical compound to its market approval, there are precisely 4 phases. In sometimes we get 1 or 2 phases combined or some extra phase, but there are precisely 4 phases. Experimental substances with the most promising laboratory results are moved into clinical trials. And during a trial, more and more information is gained about an experimental treatment, its risks, and how well it may or may not work. So this is the schematic of a phases of drug development. So just look at the left hand slider. That is the phase numbers. You can find that phases are numbered from 0 to 4, and what happens in this phase is just given in the right. So in phase 0, you have some promising substance. You find its effect on the body. If you find that it is promising, but it has some negative effect or side effect on the body, you have to step down. So you will not be able to conduct the clinical trial. Next is phase 1. So phase 1, the duration is only a few weeks, and it only involves only few patients, and it again sees whether the substance is safe for the humans. So just elaborately we can say that new treatments are first introduced to human subjects in phase 1. The primary goals include the assessment of pharmacokinetics, that is the effect of the agent on the body, and pharmacodynamics, that is relationship between the drug exposure and drug effect. And at the end of phase 1, an assessment is made to understand toxicities. If toxicity is observed to be excessive, further phase 1 studies are conducted. So these are the basics of phase 1 of clinical trial. Next comes phase 2. If your substance is good enough to pass the phase 1, then it comes to phase 2. Now look at this phase 2. The duration is more than the phase 1. That is, it involves months and it involves hundreds of patients. So the recruitment is huge. And what is the objective? The objective is to study the effectiveness at treating diseases of the substance. So we just this is just an initial study to study the activity of an agent against disease, and these trials are used for single or multiple doses, and larger groups of subjects, as you can see, are exposed to the agent in order to observe one or more clinical endpoints. So here basically drugs are compared with the existing one. So 2 important statistical concepts are introduced. So I'm not going to that details. Next is the most important one, that is phase 3 of a clinical trial. Phase 3 clinical trial involves thousands of patients. It goes around years and years, and this is a larger scale safety and effectiveness study. These are typically large randomized, I will come to this term later, to demonstrate useful clinical activity in a specific disease setting. So if a new drug successfully passes the phase 3 of the trial, the authority will approve the drug for marketing to the general people. So if your substance is promising enough to pass these 3 stages or 4 stages, it will come to the stage 4. That is, this stage is just it is called surveillance study. So in this phase are conducted only after the drug has been approved for marketing. These studies are often large and assesses a drug for undetected toxicity and side effects. And after market approval, the drugs remain under surveillance for serious adverse effects, which remain undetected in phase 1 to 3 studies. The findings may lead to the withdrawal of a drug from the market. So if you find some negative issue about the drug at this phase 4, it will lead to the withdrawal of the drug. So you will find often that some drug is being withdrawn from the market for serious side effects. So this means in phase 4, the drug shown some serious side effects and therefore withdrawn from the market. Now, what is the role of a statistician in a clinical trial? Because we know statisticians are good enough for analyzing the data. So what is a statistician's role apart from analyzing the data in a clinical trial? The most important thing is designing the trial. How do you get the data from the trial, or how do you design? So this is the basic say basic purpose of the trial, and it has to be planned by some statistician. Next is developing randomization schemes to assign patients to different treatments with an aim to reduce bias and to maintain ethics. So those people who are familiar with design of experiments in statistics or agricultural field experiment, they can just understand what is designing a trial means and what is randomization. So the idea is a little bit similar for the clinical trial also. So, and finally, the most common, analyzing and reporting the intermediate and final data. So statistician role in a clinical trial is to design the trial, develop the randomization schemes, and then finally analyzing and reporting the intermediate results. So let's see how to design a clinical trial and how a statistician can do this. So what are the different clinical trial designs? Now, based on the assignment of subjects to different treatments, we have the following useful classification. The first one is parallel treatment design, and the second one is crossover design. So what is a parallel treatment design? Say we have 3 to 4 treatments and patients are assigned to either of the treatments. So no patient receives more than one treatment. That's why the name parallel. So all this patient recruitment process for different treatments go parallelly. That's why this is called a parallel treatment design. So have the schematic, just look at this. So participants are screened for entry criteria. They are either assigned to experimental treatment or control treatment, and their responses are observed. So this is how a parallel group design goes. Now, if we have more than 2 treatment arms, the procedure is exactly same. Next is so some example. So say we have 3 treatments, A, B, and C, and we want to assign employ 1:1:1 parallel design, that is equal number of patients to different treatment arms. So for example, if you in the trial you have a sample size 54, that is 54 patients are to be assigned. So 18 people would be assigned to each of the treatments. That's how this is called 1:1:1 parallel design. Next is a crossover design. What is a crossover design? A person or treatment unit receives 2 or more study treatments in a specific order. So it's better to see the schematic. So in the left panel, you find that participants are screened. There are 2 treatments, experimental and control. So their responses are observed, and the people in the experimental treatment, after observing their responses, are further assigned to the control treatment, and the control treatment patients are further assigned to the experimental treatments. So there is some crossover between the treatments and the patients. So this is what is known as a crossover design. So in this case, each patient receives two or more treatments. So in this case, those getting the experimental treatment are also getting the control treatment, and those getting the control treatment at first are also experiment phase. So this is what is known as a crossover design. I'm just giving you one example. Here treatments are not A, B, and C. Actually, patients are assigned to the first patient assigned to control, then to experimental, then to another treatment. So there is some treatment sequences. So if you have three treatments A, B, and C, and we keep the sample size at 54, then six different orderings of the treatments A, B, and C are possible. These are ABC, ACB up to CBA. So with 1:1:1 allocation, 9 patients would be randomly assigned to receive a given ordering. So there will be 9 patients in the treatment ABC combination, 9 patients in the treatment combination ACB, and so on. So this is how crossover designs are planned. It's a bit complicated in the sense is that the responses of the patients for different treatments are correlated, because the same patients is observed for different treatments, so their responses are correlated. But in parallel design, responses from different treatments are not correlated, so analysis becomes complicated for a crossover design, but I will not go to that details. So I start from another thing, how to assign treatments to subjects. So this is the idea, because whether it is crossover trial or it is parallel treatment design, how to assign treatments to subjects, and there is some scope of selection bias. So we just start with the counter example. Assume that the investigator is able to guess the treatment assignment for the next patient. That is, the treatment to be assigned next is known to the investigator. Now, what investigator can do? Here are some possibilities. Say for the next patient, the investigator feels some treatment is best suited for him or her and assign it beating the scheduled flow of allocation. For example, suppose it was in the schedule, it was given that the 51st patient to be assigned to treatment 1. Now, the 51st patient, when comes to the clinician or investigator, investigator observes that and feels that he is best suited for treatment 2, and so assigned him to treatment 2. So that's how he is beating the scheduled flow of allocation. It is possible. Next is, suppose an investigator is working for a pharmaceutical company, maybe some pharmaceutical company worker, and obviously he is willing to see the success of the company's latest product, so can assign the treatment to the patient who is more likely to respond positively. So what does this mean? This means since I am willing to see my company's success, the success of the latest drug of the company, so I need some promising outcomes. I need some promising responses. So I find some people, they are healthy, and the intensity of the disease is not so for those people. So I assigned my company's drug to those people, and naturally, if they are not ill enough, they are more likely to respond positively. So this will give me some data which will show that my company's drug is successful in alleviating the disease. Next is, a sympathetic clinical staff tries to assign a favorite patient to the new therapy rather than placebo. Say some patient is coming to the clinician very number of times. So I have the staff is sometimes sympathetic on this patient. So there are two treatments. One is new therapy and the other is placebo. And you can recall that placebo has some psychological effect or no effect at all. So since the clinical staff is sympathetic, he will try to assign a favorite patient to the new therapy rather than placebo. So this is again against the scheduled flow of allocation. So this type of subconscious preferences results in selection bias. Each time the treatment selected is based on the subjective judgment of the experimenter rather than following the scheduled flow of allocation. So uh, these are the forms of selection bias, and if you uh, allocate patients in this manner, you will not get comparable groups. So that's why selection bias is very sensitive issue for clinical trial conducting clinical trials. So our allocation procedures or randomization procedures should be such that it is free from selection bias. Now, how to remove the selection bias? So the basic idea is blinding. So the blinding means mask the treatment from the patient as well as from the investigator. So neither the patient nor the investigator knows the treatment, but assigned it. So this means these treatments are generic, are often put in some wrapper containing some numbers 1 and 2. Say 1 and 2 if it is a 2 treatment trial. So neither the patient nor the clinician knows the exact treatment within the wrapper number 1 or wrapper number 2. So this is what is known as masking or blinding. So 3 types of trials are used. Number 1 is open label. Open label means there is no blinding issue. That is, this type of experiments are vulnerable to selection bias. Next is single blind. Single blind means the subject is blinded. That is, the subject does not know which treatment is given to him, but the clinician knows which treatment is given to him. So it is called single blind. And another thing is double blind. Double blind means neither the subject nor the investigator knows the treatments. So this protects the trial against assessment and allocation and, of course, selection bias. Achieve double blinding, or whether double blinding is always feasible or not. Actually, it would be hard to blind a patient to the therapy given in an exercise study. Say we have as treatments 2 types of exercises. So how do you blind it? It is not possible. Again, it might not be possible to blind a patient while comparing utility of different invasive procedures. So you are operating. So you are using some operating procedures, some minimally invasive procedures. So there is no possibility of blinding. However, in other cases, the best approach for blinding requires 2 groups for data processing. One group to encode the treatment data and one group to perform the analysis. So some group performing treatments within the wrappers numbered 1 and 2, and the other group performs the analysis based on treatment 1 and 2, and then finally they find which one is treatment 1 and which one is treatment 2. So these are the basic forms of double blinding. So now we come to randomization. So this shows why randomization is or deterministic allocation is not a good one for clinical trials. So we try to assign the patients in some random fashion so that no bias can arise. So randomization is the process of assigning patients to treatment using a random process or random device, such as an unbiased coin, a pack of cards, or an urn. So different forms of randomization means simple randomization, permuted block randomization, stratified block randomization, and dynamic or adaptive allocation. Now what is simple randomization? Here the trial size, that is the number of patients to be recruited in the trial, is not exactly known. Some sequential type of trial. An incoming subject is assigned to treatment 1 with probability P. This P is known. Say for 1:1 trial, your P is half. If the trial is 2:1 randomization is used, then P is 2/3. So this is how it computes. So an incoming subject is assigned to treatment 1 with probability P. So this means you are essentially tossing a coin with success probability P, and if the result is a head, you assign the patient to treatment 1, otherwise assign the patient to treatment 2. It is analogous to a coin flipping or drawing a ball from the urn. However, uh, tossing a coin or drawing a ball from the urn, these are all randomization devices and mathematical artifice. So nowadays what is done, a computer is used to generate random numbers, and then accordingly this allocation is followed. So I have some example. Say the computer generated the sequences 4, 8, up to 0. So we need to create two groups, that is assign treatment A and treatment B. So the criterion is that for even numbers you use A, and for odd numbers you use B. So this means whenever a patient comes, you find the patient number. If the patient number is even, he is assigned to treatment A, and if the patient number is odd, he is assigned to treatment B. Okay, so this is how computer generated sequence is used. So for every patient, some number is generated, and depending on the number, whether it is even or odd, treatments A or B is assigned. Now, if there are three groups, say three treatments A, B, and C, you just ignore 0, because in computer you can generate either 0 up to 9. So you designate 1, 2, 3 as A, 4, 5, 6 as B, and 7, 8, 9 as C. And for every patient, every incoming patient, generate a random number. If it is within 1, 2, or 3, you assign A. If it is within 4, 5, 6, you assign B, and otherwise C. So this is how simple randomization goes. Next is permuted block design or permuted block randomization. This is used for small studies to maintain reasonably good balance among treatment groups. With a block of 4 for 2 treatments, there are 6 possible permutations. So the idea is that we need to assign 4 patients to either of the treatments A and B. Now, what are the possible number of arrangements of two type of objects, 2 in number? That is, there are 4 objects, 2 are of some type and 2 are of some other type. So there are 6 possible permutations of this arrangement. So if we have the objects as A and B, so we have the possible permutations as A, B, B up to A, B, A, B. So these are numbered as 1, 2 up to 6. So each number, so what you do now, generate a random number using the computer. So you have the numbers 1 to 6, but in you can generate numbers from the computer generates numbers from 0 to 9. So what you do, you just discard the numbers 0 up to 9. Now if your number is 6 means you choose the possible permutation AB AB. So for the first block of 4 treatments, the treatments are assigned. Okay, now if your second number is 7, you need to discard it. Then if your third number is 1, then the next 4 patients are assigned in the form BBAA. So these are the treatments assigned to the next 4 patients. So likewise, these are done. Now the objective of this permuted block design is to strike a balance. Balance means balance within the groups. That is, you can see that in each block there are 2 A treatments and there are 2 B treatments. So whatever be the number of the patients, treatment A, the number of allocation of treatment A and number of allocation of by treatment B are exactly the same. So at the end of the trial, you find say 50 treatment A treated patient and 50 treatment B treated patient. So this is the basic idea of permuted block design. But the problem is that, say for the first code, that is AABB, since you know that each block is balanced, that is there will be 2A and there will be 2B. So if you assign 2A at the first, then definitely you know in the next part of the block the two treatments are obviously B. So this again results, there is no concealment of allocation, and therefore it results in selection bias, because whenever the clinician knows the next treatment to be assigned, there is a tendency to introduce selection bias. So this permuted block design is vulnerable to selection bias. So what we do, so the block sizes are taken as random. So random block sizes are used to just avoid selection bias. Next is allocations with covariates. So in practice, there are many covariates such as age, sex, disease history, and many others, and it influences the response. So such a heterogeneity is controlled by forming what is known as strata or blocks. For example, just look at this. Uh, Suppose you expect that there are two prognostic factors or covariates, say age and sex. So, and you expect that the response of the patients differ with respect to age and sex. So, what you have, so for the age, you define two strata, over 55 and under 55, and for sex, there is male and there is female. So we have two uh, levels of age and two levels of sex. So there will be four strata in all. And within each strata, you can either use simple randomization or permuted block randomization. So this is what is known as stratified randomization. That is randomization, separate randomization within each strata. It might be possible that in one strata you use simple randomization and in other strata you use permuted block randomization. This is possible. So next one is dynamic randomization. So dynamic randomization is something different from permuted block design or simple randomization. Uh, Actually, the essence of this dynamic randomization is to utilize the data, utilize and analyze the available data. So the idea is that at some point of time in a clinical trial, you find that some treatment is doing better. Maybe that it gives, say, 50 patients are assigned in the trial and 50 responses are available in the trial, and you find that treatment 1 is assigned 30 patients, and treatment 2 is assigned 20 patients, and among the treatments assigned to treatment 1, that is 30 patients, among the 30 patients, 25 patients cured, and among the 20 patients assigned to treatment B, only 5 patients are cured. So this definitely indicates that treatment A is doing better for the trial. So what is needed? So you need dynamic randomization or some adaptation. Adaptation means just observe the data, observe the available data, and then accordingly change your allocation strategy. So if at some point of time you find that your treatment A is doing better, for the next patient, change the allocation probability of the for the incoming patient to treatment A. If treatment A is doing better for the last 50 patients, then treatment A allocation probability would be higher for the 51st patient than for treatment B. So this is what is known as dynamic randomization or updated randomization or adaptive randomization. So here the objective is either to balance the allocation or to favor the treatment doing better. So we have some adaptive allocations. So these are some examples. So first one is Friedman-Wei's urn design. Now this allocation, this is a urn design. There are two treatments and patients are coming to the clinician and using some randomization device. In this case it is an urn, hypothetical, and they are assigned to treatments. Now how it goes, just consider an urn containing alpha white balls and alpha black balls, two colors of balls representing two treatment types. So we have two treatments. So and the urn contains alpha white balls representing treatment 1 and alpha black balls representing treatment 2. For the randomization of an incoming subject, a ball is drawn, its color noted and returned to the urn. Say you find a white ball. What does this mean? This means uh, the incoming patient to be assigned treatment 1. Okay. Now, what is the allocation probability for treatment 1? Since we have black and white balls equal in color, so for the first patient, and it is with replacement drawing, so for the first patient, the allocation probability to either treatment is half. Now, what happens after this? That's the most interesting. The corresponding treatment is assigned, I have already told you, and beta balls of opposite color are added to the arm. So actually, the objective of this Friedman-Wei's urn design is to strike a balance between the treatment assignments, that is in the long run to get equal number of patients to different treatment arms. So this is the basic objective for Friedman-Wei's urn design, and looking at that, this ball replacement possibilities are framed. Now, at some stage, you find that one treatment is doing better, not doing better, that is some patient is assigned to treatment 1. Now, what will be next? You add beta balls of the opposite color to the arm. So what does this mean? This means since you have already selected a treatment 1 type ball, so you are to strike a balance, you need some treatment 2 type ball. So what you are doing? You are increasing the probability, increasing the chance of getting a type 2 ball. So that's why you are adding beta balls of opposite color to the arm. Just in short, your first patient is assigned to treatment 1. So your treatment 1 is assigned. So to make a balance, the next patient to be assigned to treatment 2, this is what is needed. Now, this is not always ensured that you can assign the next patient to treatment 2. What you can do, you can increase the chance of getting a treatment 2 type ball for the arm. So that's why you are just adding beta balls of the opposite color to the arm. So this increases the chance of getting a type 2 ball and therefore makes a balance. So here the objective is to balance the allocation in the long run. So this is what is known as Friedman-Wei's urn design. Next is play the winner allocation. What is a play the winner allocation? If you look at the name, play the winner. So if some treatment is winner, play it. That is repeat it. So that's the basic philosophy. If some treatment is winner, that is effective, you play it or repeat it. So here, suppose we have 2 treatments. The response variable is binary and immediate, and assign the opposite treatment to the next patient if the previous patient's response was a failure, and the same treatment if the previous patient was a success. So the idea is that if the current patient's response is a success, you assign the next patient to the same treatment. That's what is known as play the winner. Since for this patient, this treatment is, that's why it is known as play the winner. And if it is a failure, then obviously you change, you assign the next patient to the opposite treatment. But the problem is that this rule is deterministic. Why deterministic? Deterministic means if you know that the patient response for this treatment is a success, the next patient's allocation is determined. If you find that say treatment 1, a patient assigned to treatment 1 and it is a success, so definitely next patient will be assigned to treatment 1. So this means your allocation is not concealed and your allocation is deterministic. And whenever there is some deterministic allocation, there is a scope of selection bias. So this rule is, though appealing and ethical, is not good enough. So there is some randomized version of this play the winner, which is known as randomized play the winner or RPW. So here the basic philosophy is the same, but the philosophy is the same, but there is some randomization so that allocations cannot be identified in advance. So here, this is an urn design. There is an urn with one ball of each type representing two treatments. A ball is drawn with replacement, the corresponding treatment assigned. So the basic idea is just before, just like Friedman-Wei's urn design. Now, if the patient's response is a success, an additional beta balls of the same type and alpha balls of the opposite type are added to the urn. So what does this mean? If the patient's response is a success, what do we expect? We expect, since the patient response is a success, so this successful treatment to be used more and more. So what you are doing? You are trying to increase the selection probability of that treatment type ball. That's why you are adding beta balls of the same type, but at the same time, uh, you don't just underrepresent the other treatment, because for this time this treatment is doing better, but for some other patient the other treatment may do better. So for this patient, if the response is a success, additional beta balls of the same type are added and alpha balls of the opposite type are added to the urn. Next, if the patient response is a failure, and additional alpha balls of the same type and beta balls of the opposite type are added to the urn. So definitely beta is greater than or equal to alpha, because you want to increase the selection probability, so beta must be greater than or equal to alpha. So this rule is referred to as RPW alpha beta. So the intuitive idea behind the procedure is to favor the treatment doing better. So this is the idea, and the same philosophy is used for the play the winner, that is uh, assign the patient to the treatment giving a success. And here you are assigning the patient to the treatment or increasing the chance of getting a successful treatment for the patient. So this is what is known as RPW allocation or randomized play the winner. So these are some references, and now I will show you something new. So what is this? Now look at this. This is the latest paper on Oxford trial. So safety and immunogenicity of the Chadox for COVID-19 disease. And here, if you look at the methods, you will find that the terms what I have used and introduced are all used here, so that you can now understand the basics of this work and get some inspiration whether to the vaccine when to come and what is going on. So just for example, just if you follow the first line, we did a phase half 1 or 2, that is phase 1 to combined single blind randomized control trial. We have already discussed what is known as single blind, what is a randomized control trial in 5 trial sites. So the trial was conducted in 5 sites in UK, and there was 2 treatments. The number one is Chadox, and second one was some main ACWY as a control. So it is a controlled, so some randomized controlled trial, and this MEN ACWY is used as a control. Then which patients are assigned, and the patients were assigned in 1:1 fashion, that is with simple randomization using equal allocation probability, that is either patient was assigned with probability half, and then some results were given, and you will find that for each site uh, some separate randomization was used. So some stratified randomization procedure was used, and then you will see the findings, and you can now evaluate whether the findings are good enough to get some vaccine in recent times or not. So with this, I conclude, and now it's time to take questions from you.